This is the fifth and last part of our virology lecture. And we're continuing with the single-stranded RNA viruses, and we're going to start out by talking about the Retroviridae family. So these are viruses that use the enzyme reverse transcriptase to convert RNA into DNA. And this family includes the retroviruses, HIV and HTLV. So HIV, or human immunodeficiency virus, transmission is through sexual contact, exposure to blood and blood products, as well as vertical transmission from mother to baby. This virus can remain latent in CD4 T lymphocytes, and HIV causes a variety of diseases. So we already mentioned Kaposi sarcoma is seen in HIV-infected individuals. There are AIDS-related diseases such as tuberculosis, a lot of different bacterial infections. There's cryptococcosis, all sorts of different diseases that are associated with HIV. So HIV is usually diagnosed serologically, and you can also do nucleic acid based testing. And HIV is treated using a variety of antiviral agents like AZT as well as protease inhibitors. Now, HTLV, or human T-lymphotrophic virus, the transmission is similar to the transmission of HIV, and it causes T-cell leukemia and lymphoma. So detection is serologically, where you screen blood for antibodies to HTLV1 and HTLV2, and HTLV1 is present in 0.025% of volunteer blood donors in the United States. HTLV is associated with T-cell lymphoma, so it's an oncogenic, causes cancer. Rhabdoviruses. So rhabdoviruses include rabies virus, and rabies virus has a characteristic bullet shape. It's the only virus that has this bullet shape to it. So that's a key feature of rhabdovirus or rabies virus that you should know. Transmission is usually through the bite of a rabid animal. So any animal can get infected with rabies and become rabid. Any wild animal, dogs, which is why dogs and cats get rabies vaccines, bats, the disease is rabies, and rabies is highly, highly fatal. You can detect it through fluorescent antibody staining, PCR, and what you would see if you were to look at infected cells is you see what's called Negri bodies, which are these cellular inclusions, which are very classic to rabies virus. So when you think of rabies virus, you should automatically think bullet-shaped and Negri bodies. So what happens is when an individual gets bitten by a rabid animal, the organism goes right into the spinal cord. It gets into the brain. It causes an infection in the brain. Individuals start to get strange symptomologies, but it could take a very, very long time for people to get symptoms. Start getting disoriented. The virus can then spread into the salivary glands and cause that frothing of the mouth. It can get into the nervous system, get into the heart. So it causes people to be excessively thirsty. So it causes a very different symptoms than most viral infections. And here is an electron micrograph of rabies with that classic bullet shape. Now, Togaviruses include viruses such as Rubella as well as Alphaviruses. So Rubella is a virus that's transmitted through the respiratory tract and as well as vertically, and causes the disease rubella. Now rubella is known as German measles. We already talked about measles, and this is a different organism. This is German measles. The problem with rubella is you can get congenital rubella. So if a pregnant mother gets infected with rubella, it can lead to stillbirth, and it could lead to a child that's born with very, very serious complications. Detection is usually serologically, and there is a rubella vaccine, so it's a preventable infection. And here's a classic German measles rash, and it looks somewhat similar to the measles rash, where you get the red spots all over the body. Now in measles virus, you get the Koplik spots inside the mouth, and there's a picture of that on your measles slide. So rubella is just the rash all over the body. Alphaviruses are transmitted through arthropods, usually the mosquito, and can cause Eastern, Western, and Venezuelan equine encephalitis. So these are encephalitis. The lining around the brain gets swollen, and detection is serologically, and you usually are going to look in the spinal fluid. When someone has symptoms of meningitis or encephalitis, immediately a spinal fluid is taken. Now, the last group of viruses we're going to talk about are hepatitis viruses. We already mentioned hepatitis A previously. Now, the hepatitis viruses are not all in the same family or group of viruses, but because they all share the same tissue tropism, they all have an affinity for the liver, we're talking about all of these viruses together. So there are at least 8 different kinds of hepatitis virus. There's hepatitis A, B, C, D, which is known as delta hepatitis, E, and G. So hepatitis A is fecal oral transmission, and it's usually through ingestion of contaminated food or water. So hepatitis A is an intestinal infection. It has a short incubation, abrupt onset, very, very low mortality, no carrier state. So an individual will get intestinal symptoms, and then it will go into the liver and cause liver infection, inflammation, jaundice, which is yellowing of the skin, icterus, which is yellowing of the whites of the eyes, and that's how you know someone has a hepatitis. And then you'd have to go look and see what kind. So hepatitis A is a little different because it is a foodborne infection, and detection is serology. So in the upper right of this slide, this is from 2005, and it's the prevalence of hepatitis A infection in the areas shaded in that kind of reddish rust color are much more high prevalence of hepatitis A. Now, hepatitis B is a more commonly known type of hepatitis. Transmission of hepatitis B is sexual or injection exposure to contaminated blood and blood products. Now, the virion of hepatitis B is called the Dane particle. That's something that's worth memorizing. Dane particle in hepatitis B. Now, the site of latency of hepatitis B is the liver, of course. It's a hepatitis virus. Diagnosed through serology as well as viral antigen detection. And the thing about hepatitis B is it has been highly linked to causing liver cancer. So, acute infection with hepatitis B virus usually resolves. In 90% of the cases, you get hepatitis B and you resolve the infection. In 1% of cases, and these are individuals that have a co-infection of hepatitis B and hepatitis D virus, that causes a fulminant hepatitis, so liver infection, jaundice, inflammation. Now, 9% of individuals with hepatitis B get a chronic or persistent hepatitis. So you can see resolution where you will not see the hepatitis B surface antigens any longer. Individuals can be asymptomatic carriers of the organism. So there's a chronic persistent infection where an individual has systemic disease, has very progressive liver disease, chronic disease, but they may not really know they have the disease until they have severe liver damage. There's also a chronic active disease, again, progressive liver damage. So clinical features of individuals with hepatitis B tend to be fever, you can get a rash, pain and tenderness in the liver, in the abdomen region, jaundice, yellowing of the skins, whites of the eyes, a low white cell count. You can have pain in the joints. Now, hepatitis C is transmitted like hepatitis B. You can be sexual contact. You can also have parenteral transmission. The disease, like hepatitis B, acute and chronic hepatitis. Hepatitis C is associated with hepatocellular carcinoma, a cancer. And just like hepatitis B is associated with liver cancer and diagnosed serologically and nucleic acid testing. Now, we're not going to talk about all of the other hepatitis viruses because hepatitis A, B, and C are more common in the United States. Now, we're going to talk about a type of organism that is not a bacteria. It's not a virus, and it's called a prion. So just because we're talking about this in the virology section, don't confuse it with a virus. This is not a virus. So prions, what a prion is a protein. It is a disease caused only by protein, in specifically a misfolded protein. So this is it's not really an organism like a bacteria or a virus. It has no nucleic acid, no DNA, no RNA, just protein. What prions cause are transmissible spongiform encephalopathies. And these are fatal neurodegenerative diseases. So you can see transmissible spongiform encephalopathies in many different types of animals, including humans. So in humans, prions cause Creutzfeldt-Jakob disease, or CJD. Now, the prion is only a protein, and it is extremely resistant to sterilization. So our viruses are they're the weakest of the organisms. They could be sterilized quite easily. Bacteria are, a little hardier. But they can be autoclaved, especially our organisms that are spore formers, like our bacillus organisms. They are much hardier. They can survive outside the body and on surfaces for many, 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 many years because of the spore, but they will be killed in an autoclave. Prions will not be killed in an autoclave. So there have been prion infections in individuals that have gotten surgery where the instruments used on them were previously used on a patient that had an undiagnosed prion disease. So even though the instrument that was being used for the surgery was autoclaved, it didn't kill the prion. So these transmissible spongiform encephalopathies, what happens is when you get infected with a prion, it alters the structure of a normal protein that's in the brain to this abnormal form. You get this accumulation of this abnormally folded protein, which causes damage to the brain. So you get nerve cell death, you get these holes almost, this spongy, sponge-like, spongiform damage to the brain and loss of brain function. So transmission is through direct and indirect contact with infected brain tissue. So if you eat certain food products that might contain brain tissue as a contamination in the processing of that type of meat product, you don't thoroughly cook it, and you can get the spongiform encephalopathies or prions. If there are areas of the world, tribes that have been known to eat brains, even human brains, that you can get prion disease that way. There is a familial form of this spongiform encephalopathies, and there's also people that get it that really don't have any known reason. So I mentioned my friend in Florida that I went and visited and ended up getting a norovirus or rotavirus. Well, one of her friend's fathers just passed away recently from Creutzfeldt-Jakob disease. They have no idea how he might have gotten it. So the properties of these spongiform encephalopathy agents, we already said, very resistant to chemicals, radiation, and heat, and can withstand autoclaving. That's very important because we commonly use an autoclave in the laboratory to sterilize things. It's not a virus. You can't really see it under a microscope, even an electron microscope. No nucleic acids. It is a proteinaceous. Prions stands for proteinaceous infectious. It does not elicit an inflammatory response. No CPE can be seen in infected cells. You will not form antibodies against it, so you're not going to serologically look for a prion. So really how prions are identified is more commonly when individuals have very characteristic symptoms, neurologic symptoms, or individuals after they die and you take brain tissue and you look at the brain tissue, you could see the classic spongy-like consistency of the brain. So this last slide is just an overview of all the viral infections and where you would find it in the body. So I know we covered a lot of viruses in one lecture, but the key thing is to know which ones are RNA viruses, which ones are DNA viruses. What are the major diseases that the viruses cause, the ones we talked a little bit more about? And if there is a key feature, characteristic, like rabies virus being bullet-shaped and the Negri body inclusions, those are things you would want to commit to memory.